Thank you to Oncologia Brazil and Jabosh for the invitation to come and share my thoughts on TRK fusion positive cancers. These are very interesting oncogenic drivers that are found in multiple different cancer types. There are two major buckets of cancers that can harbor TRK fusions. The first group is a group of cancers that are rare, where we see the frequency of these fusions is very high, meaning exceeding 90%. And those are cancers like mammary analog secretory carcinoma, secretory breast carcinoma, or congenital or infantile fibrosarcoma. And then there's a second major group of cancers that are much more common, like lung cancers and gastrointestinal cancers, for example, that in contrast harbor a much lower frequency of these fusions. Now, the good thing about these TRK fusions is that they're highly actionable in the clinic, and there are two major first-generation TRK inhibitors that have been explored for patients with all cancers that harbor a TRK fusion. These are larotrectinib and entrectinib. Larotrectinib is actually already approved in Brazil for the treatment of any TRK fusion positive cancer that harbors one of these oncogenes. And it's also approved in the US and Canada, and recently has a positive opinion from the European regulatory authorities. Just to summarize very quickly, both TRK inhibitors have high response rates. The objective response rate for larotrectinib is 81%, and the objective response rate for entrectinib is 57%. In terms of the durability of disease control, the medians for duration of response and progression-free survival have not been reached for larotrectinib, but are in the order of 10 and 11 months for entrectinib. Both drugs, we've seen intracranial activity, where the drug can result in reduction of tumors that are either primary brain tumors or metastatic tumors from an extracranial primary. And of course, in terms of the safety profile, both drugs do well and are amenable to long-term chronic dosing with comparable dose reduction rates to crizotinib, the very well-tolerated drug, incidentally with minor TRK activity, and also interestingly lower dose discontinuation rates than crizotinib. So overall, you can give these drugs for a very long period of time. They're very active and can keep patients' cancers in check very durably. Now, for some of these cancers, resistance eventually develops, and for many tumors, we found that the acquisition of resistance is mediated by the emergence of a TRK resistance mutation, or a mutation in the kinase domain of one of the three genes that make the TRK proteins, NTRK1, NTRK2, and NTRK3. The different kinds are called solvent front mutations, gatekeeper mutations, and XDFG mutations. Fortunately, we have next generation drugs. These are repotrectinib or LOXO 195. And both of these drugs are already in phase 1 clinical trials, looking at the activity of these agents that were specifically designed to address these resistance mutations in patients with TRK fusion positive cancers. And we already have proof of principle from both programs that patients who get an earlier pill, progress and acquire these resistance mutations, then jump onto the second pill on these clinical trials or compassionate use, and were able to thereafter reestablish control of their cancer. So I think it's a very interesting field. At the end of the day, the basic message here is please look for TRK fusions in your patient's cancers. The best possible test to do this, if you have the resources, is to do next generation sequencing that includes not only DNA, but also RNA. However, in the absence of resources for next generation sequencing, there are certain assays like immunohistochemistry or pan-TRK IHC that you can use as a method to screen for some of these cancers before checking orthogonally with another assay.